Hello lovelies, in this video we're quickly going to go over all the information you need for your AQA A-Level Biology Module 4, that is genetic information, variation and relationships between organisms. Now there are lots of key words in this topic that you have to be able to use properly in the exam. So take it slowly, make notes as you're going along and highlight these key words. This is a brilliant opportunity to get colour into your notes. If you want perfect definitions for all of these things, then the glossary over my website is just waiting for you to download it. There are a number of different locations within a cell where DNA can be found. In prokaryotic cells, DNA is short and circular, and it is not associated with any proteins. In eukaryotic cells, DNA is linear and long, and it is associated with histone proteins. These make chromosomes. Mitochondria and chloroplasts also have their own DNA. This is short and circular and not associated with any proteins. Similar to that in prokaryotic cells. Within DNA, we have coding and non-coding DNA. This slide is going to be basically a summary of the keywords that are needed. Please make sure you can use them all properly to show the examiner you really know what you're talking about. A gene is a section of DNA that codes for a polypeptide chain, or functional RNA. The locus is that position of the gene within the DNA. An allele is different versions of the same gene. Introns are bits of a gene that don't code for anything. And exons are bits of the gene that do code something. Homologous chromosomes are matching pairs that might contain different alleles. A triplet is a 3 base pairs that code for an amino acid. DNA has a degenerate code, so more than one triplet can code for an amino acid. The code is non-overlapping, so each base pair is only read once. The genome is all of the genes in a cell, whereas the proteome are all of the proteins that a cell can make. There are lots of words here. So what I've done is I've written you a glossary. You can see this is massive at 223 pages. I've written this because it is so important that you have and can use accurate definitions for these key words. Some of these definitions I've written for you are really long and make perfect 3 or 4 mark exam answers. But unless you use the language needed for A-level biology properly, the examiner will never know that you know what you're talking about. So the language you use and your use of keywords is vitally, vitally important in A-level biology. This is a short slide on transcription mRNA. Please remember this is just a revision video, so if you want more details on the full process, then the teaching videos where this is gone into in a lot more detail is the place to go. Now, messenger RNA is a single polynucleotide chain, smaller than DNA but larger than tRNA, helical in shape and manufactured in the nucleus during transcription but found throughout the cell. Here in blue we have our DNA backbone, and in pink our mRNA strand. The big green blob is our RNA polymerase. All of the bases will be colour coded, so green is going to be A, thymine is blue, uracil is pink, and finally guanine is black. RNA polymerase works on a section of DNA, separating the strands. Nucleotides free in the nucleus pair with their complementary base on the template strand, and the RNA polymerase moves along the strand, forming the bonds that connect the bases. forming the pre-mRNA. Only a small section of DNA is exposed at any time. Afterwards, it coils back into a double helix. When it reaches a stop signal, the RNA polymerase falls off, and the pre-mRNA can exit via nuclear pores. Splicing will occur in eukaryotes to remove introns, creating the mRNA. Translation and tRNA is another slide where you're going to see my poor animating skills. If you want more details or are confused about this, then the longer teaching video is the place to go. Here we have our mRNA and our pool of tRNAs. Translation occurs in the cytoplasm. Ribosomes attach to the mRNA chain, and the tRNAs being complementary amino acids. The ribosome moves along the mRNA, collecting more tRNA molecules. The first two amino acids are joined by a peptide bond. This is an active process that requires ATP. The ribosome can then move along to the next triplet. The third tRNA joins, and a peptide bond between the second and the third amino acid is formed. Again, the ribosome will move along, calling another tRNA, and another amino acid is joined by a peptide bond to the chain, until we have a long amino acid chain formed. 15 amino acids each second can be connected together, and there can be a large number of ribosomes acting on the same mRNA strand at once, meaning a lot of identical polypeptide chains can be produced very quickly from the same mRNA strand. This whole process stops when a stop codon is reached. Any change in the genetic code is a mutation. If it happens in genes, it is a gene mutation. These mutations can arrive spontaneously or because of mutagens. If they occur in the germline, they can be passed on to the next generation. One example is base substitutions. Here we have a sequence and the 3 amino acids that it codes for. In this second sequence, you will notice that one of each of the bases in the DNA has been substituted with something else. But because the genetic code is degenerative, these substitutions have not changed the amino acid that is being coded for. So this mutation might not be noticed. Another type is a base deletion. Here we have our DNA and the proteins that it codes for. Deletions will always cause mutations in the polypeptide chain as they change the reading frame. Here a T is being deleted. This inserts a stop codon and stops the protein early, meaning that it might not work or it might not function, be produced at all. And we have chromosome mutations that occur during meiosis. These can lead to polyploidy, commonly found in plants, where 3 or more complete sets of chromosomes are used. Or non-disjunction, where we have 1 more or 1 fewer chromosome. For example, in Down syndrome, there are 3 copies of chromosome 21, and in Edwards syndrome, there are 3 copies of chromosome 18. It is important you are very clear between the differences in meiosis and the differences in mitosis. Here is my sneaky little way of remembering. Mitosis has a T in it, so it will produce 2 identical daughter cells. Meiosis will produce 4 not identical daughter cells. These are your gametes. This is one topic where your spelling and your handwriting is very important. If the examiner cannot tell what you've written down, meiosis or mitosis, it's going to be really hard for them to give you the marks. So make sure you can spell these words and your handwriting is clear. Meiosis is two nuclear divisions, one after the other, leading to haploid cells. These gametes can then fuse together to produce viable offspring. Crossing over and independent segregation leads to an increase in variety. The stages in meiosis are very similar to that in mitosis, in that we'll have metaphase and anaphase and telophase. The DNA is duplicated, so the chromosomes get copied to become chromatids. They will arrange themselves in the homologous pairs. We will get crossing over. The chromatids will line up and are pulled to the opposite sides of the cell. The cell will divide and the new cell membrane will form and cytoplasm is divided. This is meiosis 1, followed unsurprisingly by meiosis 2. The two daughter cells will start the process of division again. They will line up just as before, but this time the chromosomes are separated, with each arm being pulled in a different direction. Here each daughter cell will be haploid and will not be identical. This is the second division in meiosis. During meiosis 1, we will have crossing over. This leads to an increase in genetic variation. Homologous chromosomes line up and then will twist around each other. Part of it will break and then will rejoin the other chromatid, leading to a mixture of maternal and paternal genes being in the same place. Another way to increase the genetic variation is independent segregation. Homologous pairs are lined up at random, so a mixture of paternal and maternal chromosomes will end up in the daughter cells. Subsequently, there are a large number of different combinations of the chromosomes within daughter cells. We can determine this by using 2 to the power of n squared, n being the number of pairs of homologous chromosomes. Now, this calculation doesn't truly reflect the genetic variety. It is an underestimate because it doesn't take into account the crossing over that occurs. The theory of evolution and natural selection explains why we have so many different types of pets and other things. Within any population, there will be a range of alleles. Not all of the alleles or individuals have the same chance of reproduction or of passing on their alleles to the next generation. Random mutations means that some individuals are more likely to survive in their environment and thus more likely to reproduce and pass on their genes. Those alleles that have provided an advantage are more likely to be passed on. Over time, this advantageous allele will become the most frequent one in the population, and other alleles will die out. These advantages can be behavioural, physiological, or anatomical. Genetic diversity is all about the number of different alleles for something there is in the population. For example, if we look at our adorable furry little friends down at the bottom here, the allele for coat length will vary for the colour, for the length of the coat. We can get short-haired cats or long-haired cats. It will vary by the texture, but these all code for the coat. Having a range of genetic diversity in a population means they're able to adapt to sudden changes in the environment. It will prevent the whole population being wiped out by a single event. Genetic diversity can be reduced if there is a genetic bottleneck. When a large number of the population die and there are only a few alleles left. It can also be small due to the founder effect, where a small number of the population splits off to become a separate population or a separate group or species. They will only take a small number of alleles with them, restricting the genetic diversity. There are two different types of selection we are going to look at: directional selection and stabilizing selection. Directional selection is where it takes an extreme adaptation to survive. This is generally due to a sudden change in the environment. For example, bacteria that reproduce after an antibiotic has been applied. Here we have a graph, and we can see that there is a low incidence of this characteristic or a high incidence of this characteristic and the number of individuals. So if we are thinking about ability to reproduce after antibiotic use, these individuals would be highly likely and these individuals would be very unlikely. In the original population, you can see we've got a bell curve with a mean at the middle. After the sudden change in the environment, only these ones here were able to reproduce. So you can see the offspring is shifted towards there being a high prevalence of that characteristic in the population. Only the individuals that can survive the new extreme environment can survive to reproduce. The population's normal distribution curve and the mean are shifted to the right in this case. Stabilizing selection tends towards the middle of the range. This will generally occur during periods without sudden changes in the environment. Birth weight of babies is one nice example of this. Here we have our bar chart with very tiny babies over here, and over this end we are going to have our very large babies. You can see that the majority of babies are clustered around the middle. If we compare that to the percentage of infant mortality in the first 4 weeks, you will see that very tiny babies are less likely to survive. This may be because the very tiny babies are the premature babies. On the other side of the graph, very large babies are also less likely to survive than those born in the middle of the range. This could be due to complications during birth, and those complications will cause the death of the baby. Here is me when I was pregnant. There is my very large baby in there because all of my babies, all 2 of them, were right over this end of the graph. Antibiotic resistance is one easy to look at example of natural selection or evolution because it happens very quickly. We can investigate this in the lab if we put a lawn of bacteria on a plate, or technicians might have done this bit for you already. Put antibiotic discs onto the plate, leave it to grow for a few days, and then we can measure the clear zones. Always good to measure it in 2 different places and work out the average of this. The closer that the bacteria can grow to the antibiotic disc, the more resistant they are to that particular antibiotic, antifungal, or antiseptic that you've put on there. There are lots of reasons that antibiotic resistance develops and is developing quicker these days. This could be the overuse of antibiotics, for example, putting into animal feed. This could be the inappropriate use of antibiotics, for example, giving out antibiotics when somebody has a viral infection. Or it could be due to the incorrect use of antibiotics. If you have a course of antibiotics, but when you feel better, you stop taking that antibiotics, that will allow the small number of bacteria that are resistant to the antibiotics to continue to grow. The increasing prevalence of antibiotic resistance is leading to a big problem. The world as a whole is running out of antibiotics that actually work, meaning that in a few years' time, the leading cause of death in the world may be due to a simple bacterial infection. There needs to be a large investment into researching new antibiotics and developing new antibiotics. However, this is expensive. For one of your required practicals, you need to be comparing the effects of different antimicrobial substances on the growth of bacteria on a lawn plate. There are a few things you need to pay attention when you do this, because if you don't do this practical carefully, it will show up in your end results. Work as closely as possible to the flame. Here I have the little ethanol burner. You might have a Bunsen burner or a little camping stove in school. This will prevent any cross-contamination getting onto your plate and growing. You need to incubate your plates for a few days, and then you need to measure the zone of inhibition or the clear zone around each of your discs. It is best to measure this in 2 different places, as the clear zone is unlikely to be a perfect circle. You can then work out an average of these 2 different things. And remember, it is important to know whether you are measuring the radius or the diameter. We can then use pi r squared to find the area of the circle, or the area of the zone of inhibition. This is a semi-quantitative experiment because you're just going to get the area. We can say that this one has a bigger area or this one has a smaller area compared to something else. We can also look at measuring the effects of antimicrobial substances on the growth of bacterial populations in broth culture. This is a quantitative experiment. Again, you are going to need to work as close to the flame as possible, flaming everything when you open it, flaming your loop to make sure you do not cross-contaminate things, to make sure the bacteria from the air doesn't get into your cultures. Once you have your cultures set up, leave them to grow for a few days. You can also spread them on a plate. You can have dilutions, and then you can actually count the number of colonies. Alternatively, you can use a colorimeter, and this will give you the percentage absorbance so you can tell how cloudy the broth culture has gone. And because of these 2 things, this is a quantitative experiment. A species is defined by being able to breed within the species to produce living, fertile offspring. For example, here we have a lion and a tiger. These can reproduce to create what is called a liger. However, a liger isn't an individual species because these are generally not fertile. Classification and taxonomy can seem like a little bit of a mouthful if you've never done any Latin. I have never done any Latin, so please excuse my horrific pronunciation. Phylogenetic trees, such as this one, can show how closely related things are. For example, we can see that animals and slime molds are actually much more closely related than animals and green filamentous bacteria. However, you would think that slime molds and filamentous bacteria would be much more closely related based on how they look. There are branch points every so often where a new species or a new section branches off from the rest of them. And if we measure these in an accurate diagram, we can see a timescale. Taxonomy is biological classification. He is one of my favourite animals again, the horseshoe crab. When it was named, it was called a crab. However, it is more closely related to spiders. You need to be able to remember the order of properly naming things. We start with the domain and then the kingdom, the phylum, class, order, family, and the genus. Finally, the species. If we compare horseshoe crabs and humans, we can see that they are both in the domain of eukaryotes. They are both in the kingdom of animals. See, I'm not even going to try and pronounce the Latin. You just need to know how to understand what it is. But we start to see differences when we get down to phylum and class. We generally refer to ourselves as Homo sapiens. This is the binomial system. It is much less of a mouthful. When you are writing this in the exam, remember to have a capital letter for your genus. If you are reading this, then the binomial system will be written in italics. There are a number of different ways that scientists can discover an evolutionary relationship. One that has always been very easy to observe before modern technology allowed us to experiment a bit more was courtship behaviour. This also allows animals to easily recognise members of the same species. For example, a peacock, a male peacock, is very easily to recognise. A female peacock, which is a peahen, is going to be very attracted to these feathers, whereas a female duck is not going to be very attracted to these feathers. People to classify these to say that these have an evolutionary relationship. However, modern genetics allows us to go further. We can look at the genomes, we can look at amino acid sequences, and we can look at immunology to come up with better evolutionary relationships and classification. Here is my favourite again, the horseshoe crab. It doesn't look very much like a horseshoe, and honestly, it doesn't look very much like a crab either. If we think about it a little bit, the horseshoe crab has blue blood. It has 10 legs. It has an armoured skeleton, so a bit like a crab, and it breathes with gills. So again, I can see why they initially thought it was a crab. Now, fossils of this are 450 million years old. So we can see that the branching point came a very long time ago. However, in 2019, it was proved using genetics that the horseshoe crab is actually a member of the arachnid family. It is more closely related to spiders than it is to crabs. Biodiversity within a community is going to be the populations of all the species in the habitat and the variety of living organisms. The number of different species can be referred to as species richness. Genetic diversity is the range of alleles within all of the different habitats around. So if we look at this example here, we can see we've got a number of different species of fish. So we've got fish species richness. We're also going to have invertebrates in here and plant life. There is going to be a range of alleles among the stripy fish, which I don't know the actual name for. And then there are lots of different types of coral, lots of different habitats all around. If we want to give an actual number to diversity, we can make a calculation of index of diversity. This is the equation that we need to practice using when we are calculating the index of diversity. Lowercase d is our index of diversity. We have the sum of, capital N being the total number of all organisms, of all species, and lowercase n being the total number of organisms of our single species. The higher the index of diversity, the more diverse an area is. If a single species is dominant in that area, then the index of diversity, the D, will be lower. Modern farming techniques can reduce biodiversity, and there needs to be a balance between conservation and farming. Here we have a picture of what could be considered a very traditional farm in the British countryside. We can see there are lots of different types of crops going on. I'm not a farmer, but I can tell that because they're different colours. Some of it is crops, and I believe these little white blobs down here are sheep. So we've got a mixture of different types of farming going on as well. In between all of these, we can see hedgerows, which are fabulous habitats for insect and bird life. And there are lots and lots of trees where, again, birds and insects and potentially deer and other small mammals live. In contrast to this, here we have a different type of farm. This only has one crop in it, and these probably all came from seeds that are identical. So seeds that are basically clones of each other. There are no hedgerows, there's no different crops, there's no animals in there. This is a very, very different habitat. There is a lot less diversity because there's a lot less animal and plant species here. Finally, here is a picture of the Amazon rainforest, or at least this bit over here is the Amazon rainforest. And this bit here, the trees have been cut down to plant a single crop field of soybeans. Now, the Amazon rainforest is a massive unexplored area of wide-ranging biodiversity. It is important that we have these massive areas of biodiversity for discovering new medicines, as an example, and keeping the diversity of species alive in there. There are animals in there that we haven't even discovered yet. However, if all of the Amazon rainforest is cut down to plant single crop fields like this, soybean, then we have lost all of that biodiversity. When we are looking at investigating the genetic diversity within and between species, there are a number of ways we can measure this, but it could be measured by observable characteristics, such as beak shape. These are influenced by a wide range of characteristics, but they can also be influenced by the environment, for example, someone's weight. We can measure it by the base sequence of DNA or of RNA, and improvements in technology are allowing us to do this more, and for us to be able to compare sequences. Alternatively, we could look at the amino acid sequence. Different DNA sequences can code for the same amino acid sequence, and this could potentially be an advantageous mutation. Closely related species will have similar DNA sequences. It is likely that you will need to do some quantitative work with diversity. Random sampling is an important technique. It would be impossible for us to measure the whole population, so we look at a small sample and estimate to find the final value. It's important we avoid sampling bias, so it must be random areas or samples that are taken. For example, here is a picture of a beach. Now, say we wanted to investigate variation in things on the beach. If we took these three samples here, we wouldn't actually get the impression there were any umbrellas on the beach, just a lot of space. However, one way you can ensure that you're looking at random areas is by drawing grid lines over your sample, and then using a dice or pulling numbers and letters out of a hat to ensure that you do get a truly random sampling going on. We can do quantitative investigations of variation. Here we have a normal distribution with the mean shown by the dotted line in the middle. The mean is the sum of all samples divided by the number of samples. The mode is the most common number, and the median is the number in the middle, in an ordered set of values. Standard deviation is how far away from the mean something is. So if we have a small standard deviation, there is not a large variation. But if we have a large standard deviation, then there is a lot of variety.